Hi, everyone. Welcome to the virtual archaeological research facility. In case I don't know any of you personally, my name is Lucy Gill, a PhD candidate in anthropology here at Berkeley, and I'm one of the brown bag organizers this semester. Before I introduce our speaker today, I will begin with a land acknowledgement modeled on the statement developed by the Native American Student Development Office in partnership with the Muwekma Ohlone tribe. We consider this a working formulation to be replaced with language reflecting the particular position of the ARF community and developed in collaboration with appropriate stakeholders. The Archaeological Research Facility sits on the territory of Huichin, the ancestral and unceded land of the Chochenyo Ohlone, the successors of the historic and sovereign Verona Band of Alameda County. We acknowledge that this land remains of great importance to the Ohlone people, that every member of the ARF community benefits from the continued occupation of this land, and that it is our responsibility to support Indigenous sovereignty and hold the University of California accountable to the needs of American Indian and Indigenous peoples. So before we get into this week's brown bag, I'm going to turn it briefly over to Sarah Kansa to make announcements.
Thanks, Lucy. I just wanted to flag really quick that next week, March 11th, is UC Berkeley's Big Give. It's an annual online fundraising extravaganza, and we are participating this year with a triple match that's been offered by 2 anonymous donors in support of archaeology at Cal. So come back to our website, and you'll also be getting emails from me before Big Give happens. But on March 11th, if you go to give.berkeley.edu/ARF, you can make a donation, and your donation will be tripled in support of archaeology.
Thanks for that, Sarah. So before I introduce our speaker today, I just wanted to say that next week we will have the pleasure of being joined by Katie Kinkoff, who will be talking about her experience as a new professor in the CSU system and her ongoing research on materializing disability justice. So please join us for that back here at the virtual ARF. Today, we are joined by Jordan Brown, who is also my brown bag co-organizer this semester, and he is a geoarchaeologist working on his PhD in the Department of Anthropology at UC Berkeley. He works primarily in pre- and proto-historic Southwest Asia, in what is now Eastern Jordan and Northern Iraq. However, he also has an abiding interest in statistical methods in archaeological data analysis and the integration of archaeological, ecological, geomorphological, and climatic data. So while I look forward to hearing about his work in Southwest Asia some other time, today we'll be hearing about a project a little closer to home, about a novel statistical approach for determining the harvesting seasonality of shellfish on the California coast. This is a collaborative project drawing on many different data sets. So towards the end of today's talk, we'll be joined, I believe, by a couple of guest stars, familiar faces to many of you, so definitely stay tuned for that. Also, this work will be presented at a session at the Society for California Archaeology meetings this Friday afternoon, along with other work by folks from the Cal Lab and the Amamutsan Tribal Band, so check that out. And without further ado, take it away, Jordan.
Thanks so much, Lucy. It's a pleasure to be here in something other than my usual capacity. And of course, do stay tuned for those special guests. You saw one of them on the screen just now. Yeah, so what I'll be talking about today is a case study in archaeological quantification, looking at shellfishing seasonality on the California Central Coast during the Holocene. And there are a lot of different aspects to this project and to the research that's related to it. I'm going to be focusing fairly narrowly on the isotopes and how to quantify them, since that reflects how I got involved in this project. But before I launch into that, I do want to just give a quick thanks to the Cal Lab for getting me involved in the first place. It's about a year ago now that we started chatting about this kind of stuff. And the SCA presentation session that Lucy mentioned has been about that long deferred. So this has turned into a longer project, which has been fortunate, as I hope you'll agree. So yeah, very, very many thanks to Kent and Rob Cuthrell and Mike Crone and Alec Apodaca for their work on this, and you'll get to hear from some of them towards the end. So why are we interested in this study here? Why are we interested in shellfish on the coast? In this case, we're talking specifically about California mussel, which are really sort of the dominant rocky intertidal species here, and very, very important in the gathering that we see throughout the Holocene, important resource for folks. Well, the sort of best single argument perhaps you can make is there are enormous shell mounds all on the coast, and shellfish feature very prominently in that. And that's sort of a, you know, a shorthand for a lot of other things that are going on with these organisms, and with people's relationships to them and the environments in which they grow, that we're going to be interested in here. Some sort of broader questions to be thinking about. Again, I'm going to be focusing fairly narrowly on sort of this as a math problem today, but we'll get hints at some of the other stuff. Is thinking about how does shellfishing seasonality, harvest seasonality, change through time? How is it related to variation in space across a region? And where, you know, how are these activities concentrated during the year? That's the sort of basic meaning of seasonality that I'm using here, not strictly in, you know, one of 4 seasons. And how does that relate to all manner of social factors, trade networks, economic structures, foodways, anything you like, really. This is pretty deeply integrated in life on the coast. And then finally, and perhaps most importantly, is you know, looking at these long-term archaeological data sets, particularly that are, you know, these are the sites and the ancestors of the folks in the Amah tribal band, and looking for, you know, lessons from those ancestors for how we steward the coast today, and particularly how the tribal members of the Amah can be involved in that, and in fact leading those efforts by looking to these long-term data sets. So I will give a brief overview of what's going on in seasonality determination from shellfish. The basic thing is that we're drilling out samples of the shell carbonate, and relying on the fact that there is a very well-studied relationship between the oxygen isotopic content of carbonate that's precipitated by mollusks, in this case, California mussel, and the temperature of the water in which that carbonate is precipitated. And so this allows us to look at temperature changes over the life of a mussel, very, very far back in time. So this is a little picture of our basic sampling scheme, courtesy of Alec Apodaca, and a little picture of a thin section as well, where you can see sort of the different growth bands. These creatures grow by accretion, one band on top of the other as they age, and so they're sort of creating a biotic calendar in a way, of sea surface temperature in the form of carbonate oxygen isotope composition. Now, I want to give a quick history of seasonality analysis using isotopic data sets, well, particularly in shellfish. And it has a long history, at least relative to the relatively young field of stable isotope geochemistry. One of the very first papers that discussed this carbonate oxygen isotope paleothermometer, as it's referred to, is from Urey et al. This is the dude who invented radiocarbon dating. So thanks, Harold. He was very, very busy. And in this paper, they sampled from the growth rings of a fossil belemnite, and I'm not sure if it's exactly a specimen, but this species became the sort of standard for isotope analysis of carbon in general. Anyway, they plot up their values and they get this nice curve and they say, we think this is seasons. That's why we see WS here for winter, summer. So that was an exciting thing to realize. And a couple of decades later, Nicholas Shackleton realized that not only was that a fancy thing for paleoclimate studies, it was also great for archaeological studies. This is a little graph from the very first archaeological isotope seasonality study, where Shackleton sampled modern shells of a particular genus or species, forgetting the name at the moment, not Mytilus californianus, not California mussel, and established sort of what the distribution of isotope values was over the whole course of the year, and then sampled some archaeological specimens and said, look, their isotope values cluster on one side of the year. This is a positive isotope value, so this is cold. I won't go too deeply into that, but winter harvesting is what he concluded. And then somewhat after that, Killingly and Berger, and then or Berger perhaps, and then Killingly, again, both geochemists with some archaeological interests, take a look at actually specifically California mussel in Southern California, and say, we think we can do one better than Shackleton. Shackleton just said this harvesting was happening during the cold part of the year. I can't say exactly when. These analyses are too uncertain. Killingly says, no, look, if we take a bunch of samples, not just a couple from the very end of the archaeological shell, if we take many, we can trace this whole curve through a long span of time. And you have to make some assumptions about how fast the shell is growing, but we feel we can do this. And then we go to our archaeological shells and say, where in this squiggle do they end up? This is where they're harvested, and made estimates of a monthly resolution on the basis of that. Shackleton didn't love this and thought that they were spending way too much money on sampling for conclusions that were far too uncertain. And in an attempt to demonstrate this, Dept and colleagues included Shackleton, took samples on the same day from shells, modern shells, and said, look, all three of these isotope profiles were harvested on the same day. Do you really think you can say to a monthly degree of resolution, when a single archaeological shell was harvested? So that's their skepticism here. I would say that there's a little bit of shape similarity in this, and Killingly's argument was about shape. So I think it doesn't fully end the discussion, as you'll see my opinion is today. So let's bring us up to the present day very quickly. The two major techniques in use are so-called terminal growth band plus X sampling, TGB plus X, which means you take the final growth band value, one isotope sample here, and then you move back at regular intervals, say 1 millimeter, 2 millimeters, 2 millimeters in our case, and you plot those values in a sort of time series. And this spacing is relatively coarse, as you can see here. And some other authors have pointed out that you might be missing important parts of the trend, important parts of the shape. And so there are authors who advocate microsampling and micromilling in order to really resolve as clearly as possible the isotopic variation in shell over time. But there's a trade-off here, and the authors that I'm mentioning recognize this, because this approach is a lot more costly and a lot more time-consuming, and it provides some benefits, but then you have a real trade-off to think about in regards to archaeological site and region sampling, because if we're trying to say something about how people interact with a resource that's all along the coast, then just getting one shell right is not enough. So that's why I am interested in the terminal growth band TGB plus X approach, if we can do a better job of understanding the uncertainties that are involved here and of trying to get useful estimates of seasonality out without sort of pre-dividing up the year into sort of bins that we can see easily in the samples. This is a problem that Shackleton confronted, and that everybody who has ever done a TGB plus X study has confronted. You're going shell by shell saying like, okay, I see a high value at the start, high temperature value once I've converted it from the isotopes. And then it goes up, but then it goes down, and I'm guessing at how much time that includes. And so I'm trying to match that up to a seasonal curve, but there's a lot of sort of expert judgment required, and that's also hard to scale if we want to do a large sample study. So what are our goals in sort of revising these methods, or perhaps trying to build upon them, is a better way to put it. The sort of king goal, perhaps, that all of these things could fall under is reproducibility. This is the concept of other researchers being able to reanalyze your data using your same methods, but perhaps varying some assumptions. And part of this is automation, making sure that it's not just you saying, here, I plotted this thing, but trying to make that an automatic process if you can, which also helps with scalability. Really trying to get a handle on the uncertainties involved, because we know there's a great deal of uncertainty. Even just the baseline we're using for the annual sea surface temperature curve in our region of interest is an important decision. We'll see that later. And then also transparency and accessibility, having, especially because this is a collaborative process in many ways, not only with other researchers, but also with the Mutsun Tribal Band, and directed towards their stewardship goals. The folks who are sort of who are our audience here are both all of these groups really need to have the ability to evaluate what we know and how well we think we know it in an interactive and sort of clear-cut way, so we don't end up with like isotopes just seeming like this is the dictate from on high, and either take it or leave it. But so this is an important thing you'll see coming up throughout. So the very basic thing that you've got to do is, when you're trying to get seasonality out of shell isotopes, is get from the isotopes to sea surface temperature, which we have an equation for, and then from distance along a shell into calendar time, which is a bit of a thornier problem. For this pilot study that I'm going to be talking about today, we make a simple assumption of a constant, on average, growth rate of 1.5 millimeters per month, which is plausible for California mussel on the central coast. But there are lots of things that I would like to look at in more detail about this later. But for now, that's what we do. And this can get you from a comparison of your archaeological oxygen isotope data on a shell to a historical reference data set of sea surface temperature, which we have thanks to the Hopkins Marine Station at Pacific Grove at the southern end of Monterey Bay. So to give a sense of the specifics of our approach, that's sort of the general things that you've got to do one way or another. But what's sort of particular about what we do is to take our historical data and transform that into simulated isotope space, which is to say that since we have this historical data set of what sea surface temperature was doing over a long period of time, we could, in principle, predict what a shell should look like that was harvested on any given day, given the assumptions of our sea surface temperature and isotope relationship and of our growth rate assumptions for this taxon. So if we do that, then we can say, okay, well, here's the temperature values that should be relevant to these different growth bands that we're sampling in our TGB +4 in this case approach. So terminal growth band plus 4 others back in time. And here's what a shell that's harvested on that day, I think it was like May 1st that I picked, what that isotope profile should look like. And we can do that for a whole database. And so we've got, May 10th. Here's your May 10th squiggle and the isotope values that you hypothesize. Here's September 11th. Here's January 13th. These are all totally arbitrary, just pulled out of the broader data set. I do this for the whole like 100-year span that we have Pacific Grove Hopkins temperature data. And as you can see, these are the different growth bands. Time proceeds from left to right. And then the next thing that you can do, which is also somewhat particular to our approach, is to standardize each of these growth profiles, simulated shell growth profiles. And say, we know that there's, perhaps like some overall, centennial trends in sea surface temperature change. We don't want those to affect our seasonality analysis. So we're going to just say we want the shape. We don't care about the specific values. So we're just getting the shape out here, plus or minus from the average. It's a very simple approach. And then we take our archaeological shell over here. I believe it's shell number 9. And we compare it to our historical database and say, does it match? Where can we find matches? And then what we do is we plot up those matches because we know the day on which those simulated shells were simulated to have been harvested. And so we can make that plot of like, here's where we find matches for shell number 9. And so we see, this histogram presenting certain peaks throughout the year. And that gives us a guess at when this shell was harvested. So then we do that for all 40 of our specimens, or potentially quite many more. This is really just the click of a button. That's that nice automation thing. And we get a sense for between these different sites. The 3 sites are SCR7, SCR14, and SMA216. SCR7 is a middle Holocene site, on the order of 6,000 years ago, 6 to 4 or so. And SCR14 is, as well as SMA216, are both late Holocene sites, so primarily over the last millennium. And all of these sites are just north of Santa Cruz. So, and this is worth noting, on the opposite side of Monterey Bay from the temperature data set and in a more open coastline environment. So anyway, this is the histograms that we see. And I submit that this is real uncertainty about when these shells were harvested. What we can then do is combine those histograms into general pictures of harvesting activity as we think, based on this very small sample size in these sites. Hey, Alec, what's going on? Well, Alec, I'll bring you and Mike on in a little bit. Sorry to give away our special guests, but if you want to hop off for the moment, you can. Yeah, you got it. All right. Zoom Pro over here. But anyway, so we see these sort of seasonal curves from the histograms. Here's if we plot a kernel density estimate over those data points, just really interpolating densities to them. And we see some trends here. Now, that's exciting. The basic takeaway here is, well, it kind of works. These might be real things, but I'll come back to that in a moment. Getting ahead of myself. So just another fun thing that you can do if you assume that if you go back to these plots and you say, well, let's take the modal value for each of these shells, the highest sort of count of matches on these histograms for our archaeological shells, then you can take that as like our best guess at the specific time of year that each of these shells was harvested. And if you do that, then you can sort of count back along this growth band here, right? And you can say, okay, well, we assume that date because from the mode of that histogram. Then we go back and we can say, based on our growth rate, how far we think that was, so we can take a guess at what this date was, and so on. And then you can treat each shell as a time series and plot up the temperature values as though you had a real little thermometer hanging out in this region of the Santa Cruz coast hundreds and thousands of years ago. What is fun to see about this is, at least for the subplots B through E, there's something that resembles actually a seasonal sea surface temperature curve. It's sort of got the right shape, which is encouraging that we're not just, that there isn't something going terribly wrong in our analyses, somewhat as an independent check on this. It's not entirely independent, but part of what makes it somewhat independent is the fact that we actually plotted this in terms of the absolute temperature values rather than those relative ones. And the curves still seem to hang together somewhat. And they're hovering around some reasonable temperature values for the most part. One thing that I will note is these 5 dots up here represent one shell, which has some very balmy temperatures for the Santa Cruz coast. That's in Celsius over here. So pretty toasty waters there, getting up close to 20 degrees Celsius. Those are off by some factor, but we actually kind of know what factor they're off by because there's this gap here. And despite the fact that the absolute temperature values, the absolute isotope values, don't match up with what we'd expect, we picked up on the shape and our analysis assigned it to a plausible time of the year, similar to these trends down here, which are, plausible absolute temperature values. One thing that Killingly and Berger did in their 1979 piece was note that carbon isotopes can also be used as a potential proxy for upwelling intensity. This is a complex relationship that I don't have time to go into, but I made the plot, and it's kind of fun to take a look at and speculate about, that this is something we could potentially try to trace also with this same data set. And there's, it's just sort of an example of how much not only information on, human interaction with the environment, but also on other environmental variables that may impact, these seascapes over the long term, how much of that sort of information is sort of ensconced in these little shells. So fun stuff. All right, so now we get to some caveats, which is, this is a mini caveat. You notice there are missing shells here. They didn't find good matches in our historical temperature data set, and that is unfortunately the nature of a limited data set. Even though it's 100 years long, there's only so many temperature conditions that you go through. It's not necessarily a balanced representation of different annual and multi-decadal cycles. El Nino variation is not necessarily, the full range of it is not necessarily included in that century. And we know there's also changes over the Holocene and sea surface temperature. So some of the shells didn't find good matches, at least within the goalposts I set up, which there is a better way of doing this. Full disclosure, I was plotting this stuff up this week. So I didn't have time to work out all the kinks yet, but so that's also why all the graphs have labels in like lowercase and saying things like day. By the way, day 300 of the year is about November 1st. So, just to give you some bearing there. Anyway, so what do you do about this? Well, you try to come up with a true functional relationship between the variables that you're interested in. Again, not something I'll go into in great depth here, but I took a hack at it. Oh, this is the real way to do it. Yeah, this is, so foreshadowing of collaborations. Well, I'll show you my version first. This is my sort of hacky way of trying to get estimates for these shells that we're missing, 2, 4, 5, 7, 10, and 23. You can get an estimate. That's great. It does affect somewhat the overall distributions, but not hugely. But the other thing that's going on here is that I'm essentially still treating each of the growth band samples as like an independent observation, which they're not, because the shape is really what we're interested in, and those shapes hang together. So this is just a sort of tag at like how you might go about this. The real way to do it is, ooh, can I go back? There we go. Is to do some fun Bayesian statistics, which is a favorite theme, but I'll leave for another time in any detail, which is to say we've got one growth band estimate here. Z is for the isotope value, and W gives us a sense of over the course of the year. And the curve here is a representation of isotopic variation throughout the year according to this temperature data set that we have, and there's noise in that curve. But if you plot a line through it at your temperature value or your isotope value, then you get regions of sort of increased likelihood. And if you take not only one, but several, for example, 5 growth band measurements, then you can start to build up a sharper estimate of when the shell was harvested. The time axis is backwards here, but it's besides the point. So moving on, plausibility. This is plausibility analysis. Do our conclusions make sense, remembering that this is a very small sample size, only 40 shells, and those shells are split across 3 different sites and 5 different sort of chunks of radiocarbon time. And so the things that we want to look to here are other archaeological data sets and archaeological models that have been developed for this region in this time period, the Central Coast over the mid to late Holocene. We want to look at historical accounts, both oral and written. What do folks know already about the way that harvesting was done on this part of the coast? That's really important information, and especially as we get into building a proper Bayesian model, this is something that we can incorporate as prior information. That's a technical term in Bayesian stuff, but basically means what do you already know about the phenomenon you're interested in, and now you're doing an experiment and adding your data analysis to that. And the thing that we'll look at today as well is the robustness of our conclusions to changing our assumptions, which is another nice thing that automation lets us do. So I'll just briefly say, since I am not truly a Californianist, though I am a Californian, but with no real expertise about shellfishing, alas. They taste good. And I'm hoping that this mathematical analysis will send me on the way to really securing a reliable source of mussels in my life. So that's perhaps a ulterior motive that I should reveal. Anyway, so I'll note very briefly that talking to the real Californianists in the room, the trends that we see taking place over the Holocene, based on that analysis that I showed you, these plots earlier. This plot scares people a little bit because it not scares, but raises questions, because it shows sort of a year-round harvesting signature in the mid Holocene, and that sort of changes in the late Holocene. That's not really what we expect necessarily to see happen for various reasons. I'll and maybe tempered by various things, but I'll let the experts talk about that. But I think what's important to mention here is that we know we're making some pretty big assumptions about growth rate and about the proper sort of reference data set to compare things with. And so those are things that I can vary in my nicely automated analysis. So that's what I'll do now. So we change the growth rate. I said 1.5 millimeters per month. Well, here are some comparisons of the conclusions and how they change for each shell if you assume 1 millimeter a month or 2 millimeters a month, which are both totally plausible growth rates, very much within the likely range of variation. And the changes are not huge. In fact, it shows up a match for the shell number 2, which is kind of nice. But there are some changes here that are relevant by a couple of months here and there that we need to account for, and certainly changes in sort of the sharpness of the conclusion, how uncertain we are about it. Now, of course, things shouldn't get more uncertain when you vary the parameters. So in fact, what you would want to do, and this is why, again, a Bayesian approach will be fun, is you can build this into the model. You can say, well, we think the growth rate is centered at 1.5 millimeters a month, but there's a bell curve of variation around it, perhaps. And then you can have that show up explicitly in your posterior distributions, as it's called, Bayesian analysis sort of goes with your prior, what you think you know already, your data, what you learned in your experiment, and posterior information, which is what you think you know based on what you knew before and now the experiment that you've done. So anyway, you can get this stuff into the sort of final density distribution. And here's the effect of growth rates on the distributions by site. Again, not huge, but not negligible. So this is something to think about. Now, another important thing to consider is our sea surface temperature data set. And we used Pacific Grove, which is, as I said, at the southern end of Monterey Bay. Granite Canyon is another recording station that doesn't have quite as long a data set, which is why we didn't use it to begin with. It's only 1975 onward. But that station is a little bit further south along the coast. So further from our archaeological sites, but perhaps in a more similar marine environment. And so it might actually be that Granite Canyon is a more appropriate comparison. This is something to investigate. But here we see this can affect the seasonality estimations by a great deal. Almost in every case, it's a couple of months. And again, this is something we would want to build into the analysis. Like, how likely do we think, we can sort of break down what we think the SST curve, sea surface temperature curve, was across the whole region that we think gathering may have been going on, this shellfish harvesting activity for a given site, right? There's lots of things that we have to think about in terms of like, how are people interacting with this specific piece of archaeological evidence that in the past, and making those things explicit in our models for this sort of analysis is really helpful. So again, this is something that we could potentially build in, but just to give a sense of the overall effect, Pacific Grove, this confusingly is different colors. Pacific Grove now, the Hopkins South Monterey Bay location that we used for our first analysis, that's in pink. The Granite Canyon data, that is the reanalysis, is in blue. And as we see there, this gives us quite a different picture, and makes us think, well, it's actually probably pretty important to get a handle on this, what the particular temperature environment was that these mussels were being harvested in, what they were growing in, because it significantly affects our conclusions, and especially given that we have this other archaeological evidence in these archaeological models that say, we should expect a different signature than the one we got from Pacific Grove, then that's really something that we need to bring in, again, sort of as prior information in that Bayesian terminology. So this is sort of the reason for taking this explicit uncertainty modeling approach, and it allows you to integrate with other data sets. So that brings me to a discussion here of where we're going with this, and in all cases that could be termed discussed in terms of collaborations. So on the one hand, we've got to do some experiments to understand California mussel growth rates on the Central Coast better. There's a pretty thin literature on that right now. Most of the studies have been done either the Northwest Coast or in the Southern California, but south of the Bight, south of Santa Barbara, which are manifestly different different ocean conditions. So we got to do some experiments, and we hope to really connect those experiments with collaborators both in the Amamutsan tribal band, working specifically so that the experiments really tie into ongoing stewardship work that they're developing around seascape stewardship. And again, we'll have Alec and Mike on to talk a bit about that, and also hopefully to work on these questions in association with the Hopkins Marine Station and the good folks from Stanford who staff it, and know a lot about these organisms and these ecosystems, and also have sort of a historical ecological interest in this stuff, but have, to my knowledge at least, not had the chance to work with archaeological data sets, and not yet had the chance to work with the knowledge that the Amamutsan have about these practices and how they tie in with sort of these social and ecological questions that go together, traditional resource management methods and that sort of thing. And another element of this further approach is modeling. I mentioned my friend Gabriel Lewis, who's a statistician over at the University of Massachusetts Amherst, and a very fine Bayesian, has been working on this problem, trying to develop that more explicit mathematical functional model that I was sketching briefly. And there are a lot of, all of the things that we talked about as important assumptions that we need to, sort of balance here. Those things all need to go into the model. And that is the wonderful potential of Bayesian statistics, is to really allow us to make clear to the world, to admit our full uncertainty without just becoming lost in limbo of not being able to conclude anything. So hoping to, I hope that Nicholas Shackleton will not come down to put the smack on my analyses here. And then thinking is always good. This is particularly thinking with archaeologists about the nature of these sites, and thinking with our tribal partners about the nature and function of these sites and places and landscape, and how that relates to our quantitative analyses here. We want to make sure that we are sampling not only the shells in a sensible way, but also the sites and the regions, and thinking about the sort of statistical properties of the analyses that we're trying to do, and the data itself and its properties, and what we're trying to say about it. So these are some important questions that now sort of having a proof of concept, I hope, of this method, that we can get into and try to build upon it. So finally, I borrowed a picture from our very own Mike Groen here. Really, the goal here is to use this knowledge from the past, and for the Amamutsan tribal band from ancestors, to nuance and inform the way that all of us folks who live along the coast here today interact with these ecosystems and with these organisms, and with ourselves in relationship to our landscape and our environments, and having these sort of detailed molluscan archives in which these important environmental variables are recorded, and then more sort of more elaborately having these detailed archaeological archives that were, constructed very intentionally by the indigenous folks, native Californians living along these coastlines, as very clear testaments to a particular way of interacting with these coastal ecosystems and resources, and managing them and reacting to changes in sort of external, perhaps, ocean climatic variables, and also conditioning those changes, buffering them, and all sorts of things like that. So this is what we're sort of all interested in getting at here, and that's very much a collaborative project. So in light of that, I will now bring on my special guests, who are very much at the core of this project that I am somewhat of an interloper in. So I just want to thank everybody at the Cal Lab here at Berkeley, and of course Ken Lightfoot for occasioning this sort of broader research theme, and also of course our collaborators at the Amamutsan Tribal Band and Land Trust for being willing to entrust these archives of ancestral practices to us to work with. And yeah, I hope for a lot more fruitful collaboration in the future. Thank you all so much.
Well, thank you, Jordan. That was an excellent presentation, and really impressive just how you've taken on the problem of isotopes and archaeological assemblages from a nuanced perspective, and contributing to a broader body of work about seasonal site use and settlement patterns and traditional resource harvesting practices, very novel approaches, and as you mentioned, very useful for the ongoing efforts of the Amamutsan Tribal Band. There's an active project right now that I'm working on as a consultant with the tribe that's integrating archaeological information such as this into efforts of restoring coastal stewardship for people who were largely removed from their traditional coastlines and shorelines. So in a lot of cases, traditional resource management practice knowledge and traditional ecological knowledge like timing of harvest, locations, extent, size, preference, and just harvesting profiles in general are largely lost in the canon of ethnobiological information that the tribe holds. So this sort of analysis can provide these key links for cultural resource practitioners who are trying to restore traditional foodways and just relationships to the coast that span millennia, but also have a very kind of seasonally prescribed means of interacting. So I think really great stuff, and exciting and very useful. To see, developing projects with more sampling, with a larger, more robust data set to just see how refined we can get these interpretations. Might pass it over to Alec Apodaca on that, because he's he did a lot of sampling for these individual shells, and see what he might about ways to refine this method.
Thanks, Jordan. Thanks, Mike. And yeah, I'll just follow up on that. I think that Jordan's call for some more experimental studies is really warranted. And the beauty about that is that with the Amamutsan Land Trust and the Amamutsan Tribal Band, going out and having controlled experiments of intertidal plots will open the door for just a lot more other things. And right now the Amamutsan Tribal Band is really interested in monitoring the effects of climate change, ocean acidification, and pollutants that are affecting a lot of the organisms on the coast. And I feel that just starting with monitoring the growth rates of mussel shells over the course of, let's say, 1 year or 2 years or 3 years, This is going to provide a lot of information into how we interpret the isotopic results once we apply it to archaeology. So, I think that, it's fruitful. We can probably sit back and feel that isotopes are fraught with a lot of uncertainty, and we can, feel that we need to tread lightly and we don't want to make interpretations. But I think the future is very bright, and we're headed in that direction.
Thanks, Alec. I know we don't have a whole lot of time left, but folks have questions, be happy to answer them, either for me or for Alec or Mike.
Kent Lightfoot, our very own, has a question in the chat, which I will ask. Can you make any interpretations about mussel harvesting for the archaeological sites at this time? And what is explicitly your next step besides eating more mussels?
Well, I'm glad Kent caught the primary aim of this talk. So, I mean, what I can speak to is what the analysis says. So, I'll go back to one of the plots here. I won't bother to do presenter view just so you see it a little more clearly. So this is sort of the basic conclusion by site and also by time period, sort of chunked up into where the radiocarbon dates associated with this context cluster. So what this says to me is that, we're seeing during most of the time that through most of the context in SCR7 that we looked at, a distinct likelihood of harvesting throughout the year. At the oldest time period, maybe a more pronounced sort of winter-spring peak. And then looking at the late Holocene sites, perhaps this is just a single, well, perhaps two shells, so I wouldn't give it a whole lot of credence. But this plot B shows SCR14, which is an inland site, which, a couple of kilometers inland from the coast. It shows a winter harvesting emphasis, and then the coastal site from the same time period, but further north on the coast, some tens of kilometers, not too much more than that, has a spring emphasis. And so mostly I think what this shows us is that we're making some strong assumptions, because of course, if we go down to the graph here, where we use a different temperature data set, that changes the conclusions pretty significantly. SCR7 in particular no longer is throughout the year as much, much lower harvesting densities in winter, and much higher sort of concentrated in spring and summer. So I think the main conclusion to be drawn here is that, we probably need larger samples. That's no surprise. We only have 40 shells and only like 15 from our biggest sites, and that's split up between these different time periods that are separated by almost 1,000 years each. So we wouldn't expect to like get the whole signature of the site in that. And then also we know that assuming our temperature data sets is the data set that we choose is important and has some pretty big leverage on the conclusion. So that's where I would leave it.
Cool. Christine also has a question. Christine Hastorf. You partially answered it. She asks, can you say if they were harvested year-round or seasonally? And then specifically asks, what about the red tide? Is that something you've looked into?
Yes. So the red tide is harmful algal blooms going on. And red tide is a natural phenomenon, or that's what it's meant to distinguish from what I've heard termed HABs, HABs, harmful algal blooms, which are not necessarily of sort of on a natural cycle. That we absolutely think is a likely reason that people would be avoiding shellfish at certain times during the year, as they become toxic to eat. And and folks would have certainly been aware of these red tides. The thing to consider is that they are significantly associated with not only sort of solar forcings in terms of kicking up photosynthesis, but also upwelling, and that's something that can vary through time. And so that's part of the reason that I showed the carbon isotopes, because that could be a way of starting to get at that. And there are also other geochemical signatures, or sclerochemical signatures in these shells that might offer potential avenues towards that. So that's a very good question. And then, yeah, I remembered Kent asked for next steps, and I would say that I'd say two next steps that come immediately to mind. One is to apply these analyses, which are all nice and automated, to existing isotope data that's already in the literature. There's thousands of isotope determinations, thousands of shells from various sites that have been recorded in a way that is absolutely digestible by this program. It just will take somebody to tabulate them and clean the data. But that would be a very quick way to bump up sample size, not necessarily specific at these sites, but over the region as a whole, and could give us a sense of what those broader patterns are. And then as a twin to that is the stuff that Alex talking about of, working with the tribe and developing these experimental plots that can also work as a sort of teaching, learning, testing ground for these traditional resource management techniques and knowledges that folks have and want a venue for. So I think those are kind of the two places to go next.
Great. Well, those are the only questions I'm seeing right now. We're already a little over time. But yeah, thank you all so much for a fun talk about a place that I think many of us are personally acquainted with. So makes it a little bit more relatable, even if we can't necessarily travel there at the moment.